Okay, it's a little after 9, so let's get started. Thanks to all of you for coming. Today what we're going to talk about is a test that I think many of you have probably used before or heard of at minimum, called the t-test. So before we get into how t-tests work, we have to revisit some fundamental concepts that we talked about last time, and also discuss sort of the theory behind how the t-test actually works. So the whole motivation behind the t-test is accounting for the fact that when we're looking for differences between groups, experimentals and controls, for example, or asking whether our observed measurement is significantly different than some reference value, that usually involves sampling. And since we usually can't assay an entire population, we have to take our measurements from samples. Ideally, as we learned last time, those samples are relatively large, otherwise there's a lot of uncertainty. But sampling always introduces some degree of certainty. If we can't actually look at an entire population, if we study 10 mice or 100 cells instead of all 100 trillion cells in the body, then there's uncertainty. And as we learned last time, there's a fundamental concept in statistics called the central limit theorem. For those of you here that were here last time, one of the fundamental insights of this is that when we take samples if we increase our sample size, the mean of all those samples that we collect gets very, very close to the true population mean. And that's true of any underlying distribution, whether the underlying data are normally distributed or Poisson distributed or follow some crazy distribution such as this one on the far left. If you take samples and compute the mean of those samples, the means of those samples will be centered at the true population mean. And the corollary to this is that if the sample size increases, say from sample size 3, 5, 10, and 20, the mean is roughly the same. The mean of the sample means is roughly the same. But what you can see is that as the sample size increases, the standard deviation reduces. And I think that's somewhat intuitive. If you take a larger sample size, the variance or the variability in those measurements gets smaller and smaller. And so this is why in biology, or really in any science, we want to take the largest sample size possible given the money that we have and the access to reagents and samples that we have. And that's not always possible given some samples are extremely precious and we can't take large sample sizes, but we want to maximize that as much as possible for this exact reason. Because essentially what we're trying to assert is that our sample, the properties of the samples that we've collected, reflect the true population of things that we're studying. We also learned that the sampling deviation, or the standard deviation of the sample means, is really a function of that sample size. So you notice that the denominator is n, the sample size. So as sample size increases, this standard deviation or the variation decreases, and that's what you see in this plot. And that's really important because, in this theoretical plot, the individual that made this plot has simulated lots of different samples of size 5 or 10 or 15 or 30 to show you that actually the properties of that distribution reflect the true population distribution. But in reality, what we typically do is we take one sample of size n, 10 or 20 or 30, and we try to write our results around that. And taking one sample tells us that the mean will be pretty close to the population mean, and the standard deviation will be small-ish if our sample size is relatively large. And so a rule of thumb that we learned last time, and this varies a little bit depending upon the exact experimental context, But our good rule of thumb is try to have a sample size of at least 30, because if you do that, what's shown on this plot is that the properties of this sample, the standard error of the mean, the actual sample mean, the sample standard deviation, get pretty close to the true population mean, which is reflected in this red line. If our sample size is really small, there's a lot of variability about the properties of the sample distribution as compared to the true population distribution, right? Okay, so this standard error of the mean, the way it's calculated, this is an important principle and what we're going to talk about next. The standard error of the mean is the sample standard deviation divided by the square root of the sample size. And that basically is an estimate of how good the sample mean that you calculated is, how precise is it. And that's really important in the context of a t-test, because essentially what we're doing is comparing means. And we're comparing the difference between means if you're comparing an experimental group versus a control. So you're asking how different is the mean of my experimental sample versus the mean of my control sample with respect to the variance in those samples. And that variance partly comes from the standard error of the mean. Okay. Okay, so has anyone, I assume most of you have used a t-test before. Has anyone actually run it? See a few nods. Yeah? Okay, so you probably know it's kind of a rabbit hole. There's a lot of different types of t-tests. And a lot of that depends upon whether you're comparing one sample to some reference value, as I'm about to talk to you about, two samples, whether those two samples have similar variance, the same variance, different variance, same means, different means. Fortunately, as I'll show you, there's a function in R that handles all that for you. It looks at the properties of the values you give it and figures out what the right test is. But we're going to start out by talking about a one-sample t-test. And so the basic setup here is we've got some observed value. Let's say this is expression. We've observed an expression for a given protein. Let's say this is a Western blot of 12. The unit really doesn't matter. But we know from the literature that the established reference expression value for that protein, say in a wild type condition, is 10. So maybe our observed value comes from some mutagenized critter, or a critter getting a different meal than a normal meal, so high-fat diet versus control diet. So basically what we're trying to ask is, does the observed expression, protein expression, differ from the reference, suggesting that the experimental conditions affected this protein's expression? So this is a one sample t-test, because essentially we're comparing our observed data to some established reference. Okay? So really, if this reference is really comes from the mean of some control set. So I have to get blood tests every year. Has everyone ever had a blood test? You know how they give you that report of like where your creatinine levels are with respect to the range? That's the same context here. We have a reference value, and we're trying to ask whether or not our value lies within sort of the expected range. Okay? So really, and I think this creatinine example makes some sense here, that mean, or the reference value itself, has a distribution, a range. There's usually a confidence interval, as we talked about last time, around normal healthy individuals' creatinine levels. That range of values establishes what would be called the null hypothesis, the distribution of the null hypothesis, otherwise known as the null distribution. So we're trying to ask whether or not this observed value lies within the expected range, okay? Or more precisely, how much of an outlier is the observed value with respect to the null distribution? And that the degree to which it's an outlier effectively becomes your p-value, okay? talk about that in a second. Well, now. So when we observe this value with respect to the null distribution, the p-value is nothing more than calculating the area under the curve of this null distribution for values that are greater than or as distant from the mean as our observed value. Okay, so it could be greater than x, and so we'd compute this part of that tail, or way less than, that far away to the lower end, okay? And that sets up another nuance about t-tests and a lot of different statistical tests, whether they're a one-tailed or a two-tailed test. You've probably heard about that before. A two-tailed test is basically looking at the area under the curve at both tails. So it's essentially saying, I'm seeing this outlier x. What I want to know is what's the probability of observing a value x that is that far from the reference value, the mean, either greater or lower, okay? A one-tailed test, rather, is saying, well, I think it's higher. I have reason to believe that the reference value should be higher given the experimental conditions, say, and I want to ask, what's the evidence, or what's the probability that any future observation will be greater than the reference value that I've seen? And so the one-tailed test in some ways is stricter than the two-tailed test because you're only investigating a particular part of the distribution. Does that make sense? Okay, so we have this concept of a null distribution, which really reflects, we assume that the null distribution is randomly or normally distributed. And that forms sort of the hypothesis, the null hypothesis, and really we're testing both in the one-tailed test, and we'll see in the one-sample t-test, and we'll see in a little bit in the two-sample t-test. Really, everything we're doing, and this is true of most, if not all, statistical tests, you're trying to evaluate whether or not your observation and the uncertainty about that observation is improbable given the null distribution. And the hard part is really just how you set up what the null distribution is. That's where all the nuance is, and that depends upon sample size and all these other criteria. But ultimately, the p-value reflects the area under the curve for the value that you observe, extreme values beyond the value that you observe with respect to this null distribution, which is sort of conveniently described as this beautiful normal distribution here. Okay. Okay, so the problem is, in this setup, we only have one observation. We did one experiment, and we did a Western blot, say, and we got an expression value for our one mouse of 12 for some protein. But we know that that's not sufficient, right? This is an experiment. There's uncertainty in that experiment. So what we actually want to ask, we want to be able to assess whether or not that 12 expression value is reproducible. How much uncertainty is there in that value? Okay. In other words, if you were to do a t-test with one observation, you would not get a very significant p-value because it's going to look at this difference between x and the reference, but it's going to say, the uncertainty about your observation is extremely high, therefore I can't trust the observed difference between x and this population mean. So what do? Yeah, you have to do more experiments, right? You have to repeat the experiment and see what the range of values that you get for x is. Okay? And essentially that's what I'm saying in this long-winded sentence here, but we need to estimate the spread of our measurement with repeated measurements. Okay? So let's imagine, so our first experiment gave us this value of 12. Well, let's say we do it 4 more times, and we see an expression value of 11.5, 10.8 it looks like, 10.5, and 9.5. Well, I actually put the values here. Okay? So if you open up R, let's just play around with this data for a second. So I'm just going to say those are our observed expression values. And we can compute the mean of those expression values. Remember, this is a sample of size 5. We want to compute the mean of the sample, right? Central limit theorem says that should get pretty close to the true population mean, although with sample size 5, maybe it could be a little bit off, right? It should be a decent estimate. Okay, so the mean of those values is 10.86. We can compute the standard deviation, which is denoted by those red error bars. That's 0.96. And we can compute the standard error of the mean, which is, remember, is nothing more than the sample standard deviation, which I just computed, 0.96, divided by the square root of the sample size. Okay. Right. So what we assume about the null distribution is that it's normally distributed, and we can actually use the standard sample standard deviation to represent the standard deviation of the null distribution. So even if the mean of our sample isn't the same as the null, we can, and I'm going to omit the theory and the math behind this, but we can basically assume that the standard error of our sample, or the standard deviation of our sample, can be applied to the standard deviation of the null. And then as we learned from the central limit theorem, as we just reviewed, we can actually, if we take the mean of our sample and the standard error of that mean, we can actually refine this hypothesis, the null hypothesis distribution, to be based upon the standard error of the mean. Because now actually we're comparing our sample mean, x hat here, to the standard error of the mean around the mean of the reference. So essentially we're asking now, is the sample mean, not the single sample value, is the sample mean fundamentally different than the mean of the reference null distribution? And since this mean, the confidence that mean is based upon the standard error of that sample mean, we're using the same standard error of the mean for the null distribution. And now we're comparing how far apart is our sample mean versus the reference mean. And the uncertainty is given by that standard error of the mean. Yeah?
So the standard deviation gives you a better idea of the spread of the data, whereas this gives you a better idea of the accuracy.
Because here we're comparing means,
right?
And the standard error of the mean, if you remember, is a measurement of how precise our mean calculation is. And that's what we ultimately end up comparing.
So I guess the question is, when you're presenting your data, is it basically just like a judgment call on your part to decide which of those you want to present?
So in the lecture, we went over maybe the last time or the time before, standard, if your goal is to compare means and to infer that differences between means can be applied to a broader population, you actually want to use the standard error of the mean, because that's essentially describing the precision of your means. Okay, everyone with me? Okay, so this is still the setup for the one sample t-test. We're sort of working through a bit brute force the theory, but essentially we're comparing our sample mean to the null distribution mean, and that's our signal. Let me jump right into this slide. So essentially this is a signal to noise problem. I've been sort of thinking more and more about these types of tests, and ultimately they are all signal to noise tests. We have a signal, which is differences between means, but the noise is the uncertainty in those measurements. And really what most statistical tests are evaluating is the ratio of that signal to noise and whether that ratio is high enough to believe that it's statistically significant. So in this context, our signal is the difference between our sample mean and the reference mean. And our noise is nothing more than the standard error of the mean, right? And since we've applied our standard error of the mean from our sample to the reference population, or the null distribution, we basically divide that signal divided by the noise. And that becomes what's called the test statistic. Almost all statistical tests have a test statistic. In the case of a t-test, t is this ratio of difference in means to the uncertainty in those means. Okay? The t statistic, however, does not give us a p-value. We're going to talk now in a second about the t distribution, which allows us to compare, essentially look up a t statistic, and knowing our sample size, we can figure out what the p-value is. Okay? So we can, I'm just extending this code here, we can compute the t statistic. I'll change this here to t stat in the following manner. Oops, I didn't compute it. Hold on a second. And we got our standard deviation. Got our standard error of the mean. And we can compute the t-stat as that difference in means. So the minus 10 that's the mean of the reference, the null distribution, versus the standard error of the mean. So essentially what we're saying is the signal to noise ratio is 2. The apparent signal is roughly twice the apparent noise. But that's not quite enough for us to get a p-value, because we have to know the sample size from which that signal to noise ratio came. If that signal to noise ratio of 2 came from a huge sample size, you're going to get a ridiculously significant p-value. But if that signal to noise ratio came from a piddly little n=3 sample size, it's not going to be very confident that that signal to noise ratio couldn't be generated by chance, right? Okay, so this is where the t distribution comes in. So this was an innovation by someone I'm going to introduce on the next slide, but the theory prior to the t distribution was these types of comparisons could be essentially looked up on a normal distribution. But the innovation was that, well, actually, the normal distribution actually underestimates the amount of uncertainty. And so the t distribution, so the normal distribution with a mean of 0 is shown in black. And then there are 3 different t distributions for 3 different sample sizes, 3, 5, and 10. And what you can see is, let's focus on sample size 3, the t distribution for sample size 3. What you can see is that the tails are fatter or much higher than the normal distribution. Another way of saying that is things way out in the tail are more probable with the t distribution from sample size 3 than would be predicted by the normal distribution. So essentially, the t distribution is a more conservative estimate of the p-values because it's doing a better job of modeling the uncertainty way out in the tails. Okay? So for a normal distribution, by contrast, you'd get might get a significant p-value for an observation like with a t statistic out here to like t=2, like for our example. But with a t distribution from a small sample size, the probability might be much higher, and therefore, since the p-value comes from the area under the curve, the p-value is going to be much higher and less significant, right? Okay. Well, that's everything there. So the t distribution is essentially a better estimate of the true noise, the sampling noise. So as I just said, the t statistic is compared to this distribution. And so essentially what we're doing, we have this t statistic that we just computed of roughly 2, we are looking up that t statistic on the distributions. Remember we talked about the probability density function, I don't know, a month or so ago? Essentially, we're just looking up the probability of, well, actually, we're looking at the cumulative density function, the probability that there is a value greater than or equal to 2 in the t distribution for sample size 5. So I conveniently chose 5 samples so we could just look that up. So essentially what we're looking at is that p-value right there. Okay. So there's a couple of details that I want to cover before we get into more t-tests. So the mean of this t distribution is assumed to be 0. Anyone have any idea? That's kind of outlined in this text, but why would the mean of this distribution be 0? That's almost. The reason it's 0 is that this test statistic is itself a random variable. And if you remember, the test statistic, I'm going to struggle with that all day, is primarily the difference between the mean, our sample mean, and the population mean. So that value can be negative or positive. And if it's a random variable, it's going to be symmetric about the population mean. So if it's symmetric about the population mean and it's computed based upon the difference, the null, most of the time, that difference should be 0, which tells you your observed sample mean is not interesting. Okay, so that's why the t distribution is centered at 0. So that's the first parameter, but it's essentially a constant. It's 0. And I'm drawing. Oh, sorry, I'll come to this in a second. The other parameter is the so-called degrees of freedom, which is a term that's thrown around all the time in statistics. And I'm not sure if everyone has intuition about that, so I thought I'd just cover it. The degrees of freedom, in the case of the t distribution, is this variable v. It's just a parameter, just like the normal distribution has mean and standard deviation. The Poisson distribution has lambda. This degrees of freedom reflects the number of values or variables in your measurement that are free to vary. So in our case, in the t distribution, the number of variables that are free to vary is the sample size minus 1. And the reason it's minus 1, and I think this is counterintuitive to a lot of people at first glance, but if you think through it, you know the mean already. You've computed the sample mean. So if you have the sample mean, only n minus 1 of the actual values that went into that mean are free to vary. Because if we have 5 observations, if we had the mean and 4 of those observations, with algebra we can compute the 5th. So it cannot vary. Right? That's the whole intuition. How many variables in our observation can vary independent of one another? Okay. So if the sample size is 5, the degrees of freedom is 4. All right, so who came up with this? Many of you probably know this story, but the t distribution of the t-test is actually more formally known as the student's t-test. It probably should be called the Gossett t-test, but Gossett worked for Guinness, and he was a master brewer, and he also trained at somewhere, Oxford or Cambridge or somewhere fancy like that, and he worked with Carl Pearson and made some sort of fundamental innovations in statistics. And his main area of interest at Guinness was actually they would plant fields of barley under slightly different conditions and try to figure out which fields had the greatest yield. And so he realized that they couldn't actually measure the yield of an entire field as their experiment, because then they couldn't make beer with it. So they had to take small samples from field 1, field 2, and actually assess whether or not the yield from those fields was better. And so he realized that there was a lot of variability in their measurements that was not modeled well by the normal distribution. hence the development of the t-distribution and the t-statistic. And he published this paper called The Probable Error of a Mean, which is exactly what we just talked about. It's a pretty nice title. Essentially, you're trying to model what's the error associated with a mean that we calculate from a barley field or a scoop of cells or a protein measurement. As you can see, the title is by student. I think Guinness, the story goes that Guinness didn't want him revealing himself because it would essentially be like a trade secret to, I don't know who Guinness competed with at the time, but Pilsner or Kell or something like that. So here's the about, but we can attribute all this intuition and the statistical test to this clever guy named William Gossett. Right, so this plot on the right, the plot on the left is what we just looked at, where we can take a t-statistic that we compute, and that's pretty simple math, and we can look up at that t-value on the right distribution to get our p-value. This plot on the right is actually zooming in to the tails, from basically here out. It's a zoom in. So now we can actually go to t equal 2 and look up n. Okay, so our n is 5. And our t-statistic is 2. So if we can look all the way up here, this is basically the point. This is t equal 2. And for the n equal 5 distribution, degrees of freedom equals 4, we can get a pretty good guess from this nice graphic that Krasinski and Attar gave us. We can eyeball the p-value. So this tells us that the p-value associated with this signal-to-noise ratio, differences in means divided by the uncertainty in those means, should be something like 0.113 or 0.115. Everyone agree? Okay. Well, it turns out that there's a simple test in R called t-test. And if we run t-test on our observed data, so this OBS EXP is our 5 measurements. The mu is the null distribution mean, okay? And we're saying we want to do a 2-sided test, as I just talked about. And guess what? It computes the exact same t-statistic as we did manually. It tells us that the degrees of freedom is 4. Our sample size was 5. And it gives us a p-value that is pretty close to what I just said because I knew the answer, but we can look it up too, right? So let's just back out and go into R and just compute this, just to make sure we all get the same result. And what's cool is R just gives you this, well, not terribly computer-friendly, but human-friendly output, which says you just ran a 1-sample t-test. The data that you gave me are found in this variable, which again is just our observed expression values. We're exploring the alternative hypothesis that our mean is not the same as the population mean. And then here's the real take home. This is what you put in your paper, essentially. Well, I guess if it's that p-value, you probably wouldn't. You'd probably do more experiments, I hope. So the t-statistic was this signal to noise. What would we do in this case if we're convinced that there is a difference and 5 observations, 5 experiments gives us this insignificant p-value? What could we do? I mean, what's really the only thing to do? Say again?
Increase the
Yeah, do more experiments, right? I mean, if you do another experiment and the p-value gets worse, you probably should stop. But you know, the only thing you really can do is try to reduce the uncertainty in your mean, right? And that's by doing more experiments. Okay. So we talked about the one sample t-test. I'd say the much more commonly used t-test, I think in biology especially, the one sample is more of a clinical test. It's usually used in a clinical context. More basic research tends to compare conditions. So in this case, we're really comparing the means and the uncertainty about those means in two different experiments. So a control population, say mice that we feed a normal diet, and an experimental condition, say mice where we feed them some awful high-fat diet and see what happens to the poor things. And that leads to box plots that look like this, where we have a median or a mean and some uncertainty about that. And we're trying to ask, essentially, are these means different, statistically significantly different? Or would the null hypothesis say, well, it's pretty, with the sample size that you have, it's quite possible to see this kind of difference. So that's what we're testing, right? And the theory behind the two-sample t-test is pretty darn simple now that we know how the one-sample t-test works. So in contrast to the one-sample t-test on the left, where we're comparing our sample mean to some well-established reference mean, modeling the noise by the standard error of our sample mean, when we're comparing two samples, we're looking for the difference in these samples, the mean of our samples, with respect to not just the uncertainty in the mean calculated for one condition, but also accounting for the uncertainty in the mean calculated for the other condition. Okay? Does that make sense? We'll get into more detail, but does that make some intuitive sense? Okay. So we're still comparing means. It's just the uncertainty is even greater now because we've got two different groups that have their own uncertainty about them. Right. So this is where things get a little bit confusing, but I'm going to try and walk you through it. So the paper in the Points of Significance series that I recommended, and if you go to Wikipedia and look this up, now all of a sudden they're talking about variance rather than sample standard deviation, which was used in the one sample t-test. The reason for that is more just in mathematical simplicity, but the other direct connection is that I think we talked about a couple months ago, standard deviation is nothing more than the square root of the variance, and the variance is nothing more than the standard deviation squared. So essentially, we end up computing the standard, a sort of a pooled standard deviation by looking at the variance in, say, the treated condition versus the untreated condition. So essentially, we start with the variance observed from condition A versus condition B, or X and Y, treated, untreated. We compute the mean of each condition, and then we adjust the variance observed for each of those conditions by the sample size, right? So again, sample size matters. The more samples we have, the more the variance is adjusted or reduced, and effectively we can have greater trust in the differences between the means of the two conditions. Okay, and this is where it gets a little hairy, but essentially what we're doing, so check it out. For condition X, the mean is like 11, and for condition Y, the mean is 10. So first thing with the signal, again, we're going to be computing a signal-to-noise ratio. The signal now is the difference between those means, which is roughly 1. But I think this graphic does a good job of really reflecting what we're trying to do. We're trying to evaluate that signal, 1, the difference in means, versus the so-called pooled variance of the two distributions. So essentially, we're taking the variance from Y, we're taking the variance from X, and we're computing a sort of a combined or pooled variance with with the intent of using that pooled variance to assess whether or not the observed difference in means is interesting or expected by chance. Okay? So let's do this for real. So thankfully, there are really nice people like Rafa Irizarry and Mike Love who put together cool data sets to show you how this stuff works. So I'm literally just borrowing data that they've put together. So they put this data set up on GitHub. It's called Female Mice Weights. If you copy these 3 lines, we're gonna read that data set that's up on the internet. It's a CSV file, and we're gonna load it into this DAT data frame. Okay, and then I can just show you what it looks like. You can see that there's 12 mice that were fed chow, which I think is just like slang for regular mouse diet. I'm not a mouse guy. And we get body weights. I'm going to guess in grams, something like that. For mice that were fed normal chow, and then we've got 12 mice that were fed a high-fat or HF diet. Okay? So I took a peek at the data, which I just showed you there. If we go to the next slide, what we're going to do is we have to load in our friendly libraries dplyr and ggplot2. And we're going to slice and dice the data a little bit. And all I'm going to do is send the data to ggplot, and I'm going to make a box plot where the x-axis is stratified by the diet that the mice received. So that's why it's x equal y. And the y-axis that's going to comprise the values for the box plot is the body weight. We're going to make a box plot, and then we're going to put the actual data points on top of the box plot so we can sort of see what drives the interquartile range and the variance and everything that is reflected in the box plot. Okay? Right? So by eye, it looks like mouse fed this high-fat diet might be significantly higher in body weight. If that's the hypothesis you're trying to test, you might be pretty excited about this box plot, right? It's like, ooh, I think it worked. The t-test is really just evaluating, given the difference in these means, the 2-sample t-test is evaluating, given the difference in these means, with respect to the total variance in this condition versus the total variance in this condition, can we trust that difference in means? Or is it likely that that difference in means could come from a null distribution with variance this extreme? And I think, as you can see, I mean, there's almost a 10-gram range in the body weights of these mouse fed the crappy diet, and it's like nearly 10 for the mice that were fed, vegan diet. Okay. So how do we do this? Let's revisit this signal to noise problem. So in the 1-sample t-test, remember we're just comparing the mean, the sample mean to the population mean, and calibrating it by the standard error of the mean that we calculated for our sample. It's really the same thing for the 2-sample t-test. But now we're comparing the difference in our sample means. That's our signal now. That's what we're trying to demonstrate. But we're calibrating that, as I said, by this pooled noise. And the pooled noise is essentially, remember we took the variance, which is sample standard deviation squared, divided by the sample size for condition A. In our case, the sample sizes are the same, but there might be situations where you tested 20 high-fat mice and could only do 18 control mice. So that's why we have 2 different ends, those 2 different conditions. We sum the variance in those 2 conditions, and then we take the square root of that. And remember, the square root of the variance is standard deviation. So essentially, we're taking a pooled standard deviation. And that's our measurement of sort of combined noise in the experimental system. Everyone with me? Yeah.
Is standard deviation not additive the same way that variance is, or could we do this?
Variance.
Just with standard deviation?
Yeah, so the theory that I'm outlining is if you take when we're taking differences in means, you really want to like compare the difference in variance too. But actually what you end up doing, and this is theory I omitted because it's a little difficult for me especially, but you can actually add the variances. And when you add the variances, you can take the square root to get a combined standard deviation. Why you can't do it directly from the standard deviation is not entirely clear, but actually Rafa Irizarry's blog post goes into extreme detail on that. Yeah.
So a question about this one, you're comparing like the chow and the high fat.
Yeah.
So is this essentially assuming that the population mean isn't going to change between those two groups of mice? Or it seems like you're just subtracting like the T from chow minus the T from the high fat, and then the population mean just kind of disappears. Is that sort of?
So, I mean, the population, there's two population means. There's the true population mean for all mice that could ever be fed high-fat diet, and there's the true population mean from, and so we can't measure those. We're just measuring samples from those populations and comparing the means from those samples as a proxy for the difference in means for the combined, each full population. Does that help?
Yeah, no, that makes sense, I think.
Okay.
Yeah.
Okay, so again, it's a signal to noise problem, and we get a t statistic, right? And the beauty is now we can take that same t statistic and degrees of freedom, which gets a little more complicated in this case, and look it up on that distribution, right? I mean, I think conceptually we're doing the same thing. We're computing a t-stat and we're looking up on a distribution. The trick is, in this case, it's a little harder to think about exactly what t distribution we look up, because remember, the only parameter that varies for the t distribution is the degrees of freedom. Okay, so we'll come back to the degrees of freedom in a second, but we can actually test, let's figure out using R whether or not these are statistically significant differences in means of the two populations. So I'm just going to use dplyr to subset our main data set into two smaller data frames, one that's the mice from the control diet, one from the high fat. Okay, then, I'm going to compute the difference in the means. So that's pretty straightforward. We take the mean of the body weight variable for the high fat data frame, we take the mean of the body weight variable for the control diet, and we compute this basically the numerator of that t statistic. Okay. Then, as Tom just pointed out, theory tells us, and theory that I don't fully understand, but we'll read up on hopefully when I get some time, that the variance of the difference of two random variables is actually the sum of the variances. So we can compute the variances separately and take the square root of their sums. And that becomes this pooled noise. Okay. Right. So the t statistic is now just that difference of means divided by the noise, which I'm showing here. Okay, so let's see what that is. So it's a little above 2. In the one sample t-test, we had a t-stat that was pretty darn close to 2, but the degrees of freedom was low, right? It was 4. Our sample size now is 12 for the control, or 12 for the normal chow, and 12 for high fat. So perhaps our degrees of freedom is much larger now, because it's, it might be something close to 24. And therefore, that same t-stat with higher degrees of freedom, or higher n, essentially, might be a much more significant p-value, right? Because we knew that the lower the sample size, the higher the tails were in that t-distribution to reflect the uncertainty about small sample sizes. So maybe this t-stat is enough. Okay, so we get a t-stat of 2.05. And this is what I'm getting at. Like, now what do we do? Right? How do we look this up? We know it's 2, but where do we go on the y-axis? Where do we stop on the y-axis? Okay, well, let's just cheat and use the t-test function again. It is the exact same function name. It's just in this case, you give it two vectors of observations. One, the weights from the high fat, and the other, the weights from the normal. And it figures out the fact that since you gave it two lists, you're trying to do a two sample test. Okay, so we get that same t-value, t-statistic that I just computed manually, but we get this odd degrees of freedom. It's a fraction, it's a sort of float. So why is that? The answer is it's complicated. So the degrees of freedom is actually close to n-1 plus m-1, which would be sort of the intuition, because for the one sample test, the degrees of freedom was n-1. So we would naively guess maybe it was 12 minus 1 plus 12 minus 1, which is what, 22? Yeah, 22. But it's actually 20.236. The reason for this is the degrees of freedom is also pooled in this case. And it's weighted, I guess a summary of what you can read here, which I don't advise unless you really care and are interested in being bored. But basically what you're doing is you're weighting that degrees of freedom by the variance in the two samples. Variance. And so it gets downweighted a little bit from 22. We're reducing our degrees of freedom a little bit based upon the observed variance in our data. Okay, but we get, I guess intuitively, a p-value that is a little more improbable or significant, given that we have more degrees of freedom than we did in the one sample t-test for the same degree t-statistic. You know, sort of, I think Mike and Rafa chose this data very carefully, so we get this borderline p-value. So this is a bit of a conundrum. What the hell do you do with 0.053? This is where you get things like trending towards significance or plausibly significant, or I don't know what other ones are. If anyone, there's a lot of jokes about this. And this is getting at sort of, you know, maybe something we'll talk about later, you know, p-hacking and how can you maybe manipulate things a little bit so that you get over the finish line of 0.05. I'm not advocating that. I'm saying that's what's often done. And we'll also have in a future discussion talk about how the 0.05 threshold even came to be, and when not it's relevant. So we know that our degrees of freedom is roughly 20. So all we do is we take this test statistic and we look it up on this lightest blue shaded line, which reflects sample size of 20. I know it's not quite right, but I didn't want to make this plot myself, so I'm just borrowing the one that's in this paper, and that's pretty darn close to the p-value that we get, 0.053 from this two sample t-test. Okay, one other thing I want to mention is this two sample t-test is not called the student's two sample t-test or the Gossett's two sample t-test. It's called the Welch's two sample t-test. And this is where you can go on a horrific rabbit hole on the interwebs about all the different flavors of t-tests depending upon whether it's the same means, same variances, different variances. And it turns out that the Welch's t-test is best used, especially when the variances between your two groups are different. Okay. Right. So what could we do? So we're on this threshold of 0.053. How could we get it over the finish line, so to speak? Well, honestly, what you'd want to do is do more experiments. And so I did a in silico experiment where I just added 2 more weights, 1 more. So now we have 13 samples for the high fat and 13 for the control. And I gave them weights that support the alternative hypothesis artificially. So I made the high fat weight higher than the control weight. And I'm just using this R-bind function I don't think we've learned before in R, but that allows us to tack on a new row to a data frame. Okay? And we have to give it a list. Normally you give it C, but if you use C instead of list, this number gets turned into a, not a number, it turned into a factor and it breaks the t-test. So you have to use this list function. But if I do that and tack on these two more cheap experiments, very, very cheap, what we can see okay, so now we have 13 observations, right? Instead of 12. We run the t-test again, and boom, we artificially hacked our p-value with a free experiment. I just manipulated an experiment. Probably would lose my job if I published this. Should. But if we were to do something like that, the whole point is that if you do more experiments, you can increase the degrees of freedom, and ideally, if the variance in your experiments is reduced and you have better confidence in your means, the signal, and better confidence in the noise, essentially the pooled variance, then you're going to get a more significant p-value because you've increased the certainty that those differences in means are true and not driven by random chance. Okay? So the one aside, we'll come back to this stuff later, but you've probably heard of ANOVAs, or analysis of variance. Hopefully now you have a sense of why it's called that. ANOVAs are really t-tests applied to 3 or more groups. So you've got your 1 sample t-test, you've got a 2 sample t-test, and ANOVAs are 3 samples or more. And the acronym, or I guess it's a portmanteau of some sort, but is driven by the fact that you're comparing the means across multiple groups, but analyzing the variance to assess whether or not those observed means are real. Okay, and when we get into linear models in December and January, what we'll also see is that the t-tests and ANOVAs are actually just special cases of linear models, of regressions. And I think, just to say that, now that I've said that out loud, I don't know that I meant to, what you can kind of see is that you might be fitting a regression line between these two cases, but acknowledging the fact that the slope and the intercept of that regression can vary a lot given the uncertainty in the measurements. Okay, so that's the connection to linear modeling. All right, so my desire is at the end of Thanksgiving, the end of November, on the Monday before Thanksgiving, I'm going to give the next talk on power calculations and how that is driven by sample size. If you've ever written a grant or gotten a bad review on a paper, sometimes reviewers say, well, your sample size isn't large enough. You need to do a power calculation to figure out how many samples you need to observe a significant p-value. So we're going to talk about all the fun of that. But there is I'm putting it next in this lecture series because there is a really strong connection to what we've learned today. That is in many ways driven by variance, the variance and the uncertainty in our observations. Okay, so with that, we're good, and we've got 15 seconds left. So thank you for coming.